Okay. Uh, uh, so let's start one more chapter. Its chapter name is called Amino Acids and Proteins. So, protein. Okay, let's give an entry. Okay. Uh, so amino acid proteins. Proteins are the polymers. Okay, more particularly polyamides having a three-dimensional structure uh, are called proteins. And for these proteins, the monomers are amino acids. So let's give an entry. Okay. So amino acids are the building blocks of proteins. So with that, we'll go for a introduction part, and it's also comes under CH202. It's a paper code, and the paper name is organic chemistry. The chapter name is amino acids and proteins. So here, what is the relationship between the amino acids and proteins? Means amino acids are the monomers for the polymers called proteins. Okay, with this, we'll go for a introduction part of amino acids and proteins. Okay, so then proteins are the most complex materials. Proteins are the most complex materials produced in nature. The name protein is derived from the Greek word protease, meaning of prime importance. Proteins are the basis of protoplasm and are present in all living organisms. Without proteins, life would not be possible. Proteins perform several functions in the body. As hormones, they regulate metabolic process. As enzyme, they catalyze biochemical reactions. As antibodies, they resist and nullify the effect of toxic substances. Proteins are present in muscles, skin, hair, and other tissues that made up of non-bony structures. So with this, I will go to the next slide. So chemically, proteins are high polymers. Chemically, proteins are high polymers. They are polyamides. Maybe you can correct it this as a they are polyamides. The monomer from which they are derived are amino acids, more particularly alpha amino acids. So alpha amino acids means both the carboxylic acid and the amino group is attached to the same carbon, and that carbon is called alpha carbon. And that is the reason these are called alpha amino acids. Here, acid is the parent, amino is the substituent. Amino, right. Amino group, it is attached to the alpha carbon. So that is the reason it's called alpha amino acid. A single protein molecule, a single protein molecule contains hundreds or even thousands of amino acid units. A single protein molecule contains hundreds or even thousands of amino acid units. So you know that 20 amino acids are there. 20 different amino acids arranged repeatedly. 20 different amino acids arranged repeatedly in a number of ways and form different proteins in living system. The number of different proteins molecules that are possible is infinite. The number of different proteins molecules that are possible is infinite. Thousands of different proteins are required. Thousands of different proteins are required to make up and run an animal body. And this set of protein is not identical with the set of protein required by an animal of different kind. So proteins have very high molecular weight. Their molecular weight may range from 10,000 to over 50 millions. That means high molecular weight. All proteins yield alpha amino acid upon complete hydrolysis. All proteins yields means give alpha amino acid upon complete hydrolysis. These proteins may be defined as the higher molecular weight organic material which have, so that means this will be the definition. So definition of proteins means proteins may be defined as the high molecular weight organic material, high molecular weight organic material which upon complete hydrolysis yield amino acid, which upon complete hydrolysis give amino acids. So that is the definition for amino acids or proteins. Proteins may be defined as the high molecular weight organic compounds or materials which upon complete hydrolysis gives or yield amino acid. This will be the definition for proteins. So definition for proteins means proteins are the high molecular weight organic compounds or materials which upon complete hydrolysis give amino acid. Let's give an entry. So with this, I will go to the, yes.
Sir, the chapter name is amino acids and peptides. No, sir.
Maybe peptides, polypeptides means is nothing but a proteins. Okay, so
Protein is
the
more
difference
than
between 1 minute, the difference between the polypeptide and proteins. So polypeptides having three-dimensional structure, one is called proteins. Clear? Maybe I will change the heading. Okay.
but
So
the amino
initially it was means when the CBCS is started, no, more particularly before trifurcation, the name of this chapter was proteins. So now maybe since the BCU maybe changed the name. Anyway, I will uh, change it to a polypeptide. But you should aware that the difference between the protein and the polypeptide. So polypeptides having high molecular weight and having three-dimensional structure, one is called proteins. Clear?
yes, sir.
I will change it, no problem. As per the syllabus, I will change it. Fine?
Okay, sir.
Right. So with this, I will go to the amino acids, more particularly. Now onwards, I will talk about the amino acids. This is all this information is about introduction of proteins. So I will go to the next slide. Next slide will give you the information about amino acid. So What are amino acids? So amino acids, alpha amino acids. So in this chapter, if I am talking about amino acids, means it should be a alpha only. Alpha amino acids are the building blocks of proteins. Alpha amino acids are the building blocks of proteins. Amino acids are the compounds which contain amino group. Amino acids are the compounds which contain amino group, NH2 group, and this is A and D, and carbonyl group. Carbonyl group means COOH group attached to the alpha carbon atom. Attached to the alpha carbon atom or attached to the same carbon atom, that is called alpha carbon. So what it means? It means that alpha amino acids are the building blocks of protein. Amino acids are the compounds which contain amino group as well as the carboxylic group attached to the same carbon atom, and these compounds are called amino acids, more particularly alpha amino acids. All amino acids have the general structure. Let's give an entry. Okay, let's not give an entry. Okay, all amino acids have the general structure like this. So that means this is the carbon atom. To this carbon atom, amino group is attached and also a carboxylic acid group is attached. And this will be the H. And here I am written as a R. And for all amino acids, this NH2COOH, H is common. Okay, this part. Okay, this part is same for all amino acid, but only the changes in the R group. R may be a hydrogen, R may be a CH3 group, R may be a phenyl group, phenyl in the sense benzyl group, R may be a isopropyl group, R may be a isobutyl group. What it means? R may be a hydrogen, R may be a alkyl, R may be a quite complex containing other functional groups. So we will this is about information about alpha amino acids. So amino acids means it contains carboxylic acid as well as amino group. It will have a general structural formula like this, and this part is same, only the changes in the R group. R may be a alkyl group, R may be a hydrogen, R may be a quite complex containing the functional group containing compounds. So with this, I will go to the classification, more particularly amino acids classification. So classification of amino acids based on the number of NH2 and COOH groups. Based on the number of NH2 and COOH group in amino acid molecules, the amino acids are classified into neutral amino acids, acidic amino acids, and basic amino acids. So let's take the neutral amino acids. This neutral amino acid contain equal number of amino and carboxylic group. So that means maybe what I can say, one amino and one carboxylic acid containing amino acids are called neutral amino acids. So I will give an examples. So here you need to remember naturally occurring amino acids. There are only 20 amino acids. From that, I will tell you what and all belongs to the neutral uh, amino acids. I will explain in the next slide about the name of the amino acids and the symbol used for the amino acids and also structure of the amino acids. So this information I will give you in the next slide. So I will go to the next slide. So this will be the serial number 1, 2, 3, and the name of amino acids and the symbol used for the amino acids, and this will be the structural formula of the amino acid. So you should aware about the structural formula of the amino acid, because they will ask you to write the glycyl alanine peptide. Then you should be able to write the structure of glycyl alanine peptide. Anyway, I will tell you what is peptide, how the peptides are formed, all those things once the uh, discussion about the amino acids and their synthesis is completes. So first one, a glycine. So what is glycine? Glycine is an amino acid, and more particularly, it contains one amino group, one carboxylic group, and both the side hydrogen is there. And this is the GLY is the symbol used for representing a glycine amino acid. And you can remember that since glycine contains both the side hydrogen, this is the only amino acid which is optically inactive. So rest of the amino acids are optically active. So that means optically inactive amino acid means it should be a glycine. Okay. This is the structure of glycine. And let's go to the second one. It's called alanine, and the symbol used for this ALA. And here is the changes. Here in the glycine, R is equal to H. In alanine, R is equal to CH3. So what you can observe, there is a changes in R group. So when R is equal to CH3, alanine is the compound. So alanine is again, it's a optically active compound. So again, optically active means you should aware that it can be a dextrorotatory or it can be a levorotatory. And one more thing, what you can observe that if you are looking into the configuration, okay, configuration means with respect to the when configuration given to configuration is a theoretical phenomena. It is assigned based on the arbitrary standard glyceraldehyde. So the priority group, so NH2 is the priority group, means nitrogen is the priority group. If the priority group is on the left hand side, then it's called L configuration. And one more thing you should remember that almost all, okay, all amino acids, more particularly naturally occurring amino acid and physiologically active amino acids are belongs to L form. Okay, except one is there, that is 21st amino acid, that I am not talking now. But as per the uh, uh, there are only 20 amino acid. Of course one exceptional 21 also is there, that I am not talking. And out of 20, all amino acid belongs to L configuration, and L configuration ones are biologically active, D ones are inactive. This much information you should aware. So then it's about alanine. Then phenylalanine. Phenylalanine means CH2 phenyl containing amino acid is called phenylalanine. CH2 phenyl means benzyl group. Benzyl group containing amino acids are called phenylalanine, and the symbol used for PHE. Serine. Serine means it's an amino acid, more particularly CH2OH containing amino acids are called serine, and here the symbol used one SER. And valine. So valine means isopropyl group CH, CH3, CH3. Isopropyl group containing amino acid is called valine. Okay. So with this, I will go to the next slide. Next slide also will give you few more neutral amino acid, neutral amino acids. So I will go for that. So leucine. Okay, here isobutyl group CH2, CH, CH3, CH3. Isobutyl group containing amino acid is called leucine, and the symbol is LEU. Isoleucine. So isoleucine means instead of CH3 group here, CH3 will go to this position. So this is isoleucine. So that means CH, CH3, CH2, CH3 containing amino acid is called isoleucine. The representation, see, symbol for isoleucine is ILE. Then cysteine. So cysteine means the what is called as symbol is CYS, and the structure is CH2SH. That means sulfur containing. Okay, sulfur containing amino acid is called cysteine. Methionine. So methionine, its symbol is MET, and you know that CH2SCH3. Okay, this group is called methionine group. CH2SCH3 containing amino acid is called methionine. Tyrosine. Tyrosine means you know that this is a phenol. Okay, OH benzene. Phenol containing group is called tyrosine, and the symbol used for tyrosine is TYR. And with this, I will go to the next slide. The next slide also will give you few more examples of neutral amino acid. Tryptophan. So tryptophan means this particular group is called indole group. Okay. So indole containing amino acid is called tryptophan. And the symbol used for this is TRP. Okay. TRP means tryptophan. And then threonine. Threonine means CHOH CH3 containing functional group containing amino acid is called threonine, and the symbol used one is called THR. And then glutamine. So glutamine means the symbol is GLN. Okay, GLN is called glutamine. So this is also a neutral because it's an amide. Okay, amide is in neutral condition. And here what is the thing is important? Uh, CH2, CH2, CONH2. And what we can say that amide functional group. Amide functional group containing amino acid is called glutamine, and the symbol is GLN. And then proline. Okay, proline is an amino acid. The symbol is PRO. And what is the structure? You can observe that it's a pyrrole. And here this carbon is alpha carbon. This carbon contains amino group. It's a secondary amine. And this carbon also contains carboxylic acid group. The only cyclic amino acid. This is a cyclic amino acid. Okay. Heterocyclic, you can say that. Heterocyclic containing amino acid is called proline, and the structure is this. So all these examples are the neutral amino acids. That means out of 20 amino acids, 15 belongs to neutral amino acids. Another two for acidic.
Excuse me,
Another
sir.
Yes,
Sir, proline is optically active or inactive, sir?
proline is optically active because it is having a chiral carbon. This is chiral carbon only, optically active.
Only three groups are there now, sir, for chiral carbon.
Proline also optically active because what is the criteria for optical active? Optically active criteria is the carbon should attach a 4 different groups. So this particular
Then only
carbon,
three is there now, sir, here.
no, one more is hydrogen, no?
Yes, sir.
Then it should be a 4 different groups are attached to this carbon. This carbon only the chiral carbon, and this exhibits in plus and minus, means dextrorotatory as well as levorotatory, and this also exhibits in D and L configuration, and it is a optically active amino acid. Any doubt?
No, sir.
Okay, so it is a optically active, but generally it's called as a heterocyclic containing amino acid, means proline. Okay, right. So with this, I will go to the next slide. So here I am taking the acidic amino acid. So acidic amino acid means uh, it contain one NH2 group and two carboxylic acid containing amino acids are called acidic amino acids. The example is aspartic acid. So aspartic acid contains two carboxylic acid and one amino group. And the symbol used for this is ASP, is called the aspartic acid. And one more is glutamic acid. So glutamine is different, glutamic acid is different. So glutamine is a amido containing amino acid. Glutamic acid is the acidic amino acid, and the symbol for glutamic acid is GLU. The symbol for glutamine is GLN. Okay, that is the difference you need to remember. So these two are the examples of acidic amino acid. So with this, I will go to the next slide. Next slide will give you an information about basic amino acid. So this is lysine, okay? Leucine is leucine. Leucine is different from the lysine. So lysine means you can observe that a long chain amino group containing long chain means butylamine 1, 2, 3, 4. So butyl amino group containing amino acid is called lysine. So this, if any protein contains lysine as a side chain, means this that protein will act as a base. This amino part can be used. I think you are aware about that. What is called biological importance of vitamin E. So the protein opsin uses the side chain, okay, side chain of lysine, okay, this one. This NH2 is used to bond with the double aldehyde present in vitamin A. So the obtained one is called activated complex, otherwise activated rhodopsin. So this NH2 used there in the double bond formation with aldehyde. The protein opsin uses the side chain amino group to form a activated complex with the vitamin A. Okay, so uh, the symbol used here LYS. Okay, LYS and leucine. Then one more amino acid, you can observe that this group, okay, NH, double bond CNH, and then NH2. This group is called guanidine group. GUIDINE, guanidine group, guanine group or guanidine group. Guanidine group containing amino acid is called arginine. Okay, I think the guanidine containing derivative is one anti-diabetic drug. It's called metformin. I think many people are used to take for diabetic patients before food. That is metformin. In that, guanidine is the one functional group present. Then, guanidine containing amino acid is called arginine, and the symbol is ARG. And this, I think you are aware that this moiety is called imidazole. So, imidazole containing amino acid is called histidine, and the symbol is HIS. So these are all the examples of amino acids and their structure and their symbol. Okay. So with this, what I will do, I will go to the next slide. So next slide will give you the little more information about amino acids. Okay, more particularly a special category. So I'm while discussing amino acids itself, I mentioned about the special categories, but I am showing it in a one more slide. You just look into this. So what you can observe here, a special category of amino acids. So optically inactive amino acid is called glycine. Optically inactive amino acid is called glycine. Aromatic amino acids are phenylalanine, tyrosine, and tryptophan. So tryptophan means indole is there, tyrosine means phenol is there, phenylalanine means benzyl group is there. So phenolic amino acid means tyrosine. Okay, phenolic means phenol containing amino acid is called tyrosine. So heterocyclic amino acid means tryptophan. Indole group is there. Histidine, imidazole. Yes, imidazole. Yes, imidazole group is there in the histidine. And imino group. Okay, imino means secondary amine. Amino acid containing one is proline. Amino as okay, acidic amidic means amide containing amino acids are asparagine and glutamine. So these are the few special categories of amino acids. So now what I will do, I will go to the synthesis. Okay, synthesis of amino acids. So totally there are 7 methods for the synthesis of amino acids. Okay, I think this synthesis of peptide, synthesis of protein takes a wide application nowadays. Okay, not only nowadays, the year 1970 onwards, this field is bloom. Okay, so then I will go for a synthesis, more particularly what are the methods available for the synthesis of amino acids. So after the synthesis, after discussing the synthesis of amino acids, I will take up the peptides. Okay. So now let's move on to the synthesis of amino acids. So this is one method, okay, by the amination of alpha amino acid. So amination means NH2 group is alpha halo acid. So alpha means it's a carbon, adjacent carbon. Alpha halo. So here I am taking an example. This is a carboxylic acid, and this carbon is called alpha carbon. And this halogen, okay, in of course here it's a chloro. Instead of chloro, if we are taking a general halide, X means, then we can say that alpha halo acid. So this will be more particularly, this is called alpha chloroacetic acid. But if you want to say generally, if X is equal to X, then we can say that alpha halo acid. So alpha halo acid is treated with ammonia. What will happen here? Amination. So, and one more thing, I think you are aware about the nucleophilic substitution. So NH3 is a nucleophile. It will attack this carbon, and Cl is a living group. It will go out. So HCl will go out. So there will be a new bond formation between this nitrogen and this carbon. And you know that this is a acid. So ammonia is a base. So it will form a salt. So what will happen? Alpha halo acid, when it is treated with ammonia, it forms a amino acid ammonium salt. This ammonium salt, when it is treated with the proton source, more particularly on acidification, gives glycine. Okay. Since you are taking alpha chloroacetic acid, glycine is the obtained. If you are taking a different one may be a, suppose if there is a CH3 here, okay, here is there. So this is also a bromo, alpha bromo propionic acid. Alpha bromo propionic acid, when it is subjected to ammonialysis, means ammonia NH2 group is added, Br will go out. It's a nucleophilic substitution. The obtained ammonium salt is hydrolyzed to get a alanine. So here glycine as well as alanine is synthesized by this method. It is also possible to synthesize valine. It is also synthesized to possible leucine. It is also possible to synthesize aspartic acid. Suppose if you want an aspartic acid, instead of CH3, you take CH2COOH. Then the obtained one is aspartic acid. So in place of H here, if we are taking isopropyl group here, you will end up with valine. So here also isopropyl, here also isopropyl. So overall, the first method of synthesis of amino acid is by the amination of alpha halo acid. So if you want to write in terms of words means, you can write that uh, you can write that, so alpha chloro or alpha bromo carboxylic acid react with excess of ammonia to form ammonium salts of an amino acid, which on acid hydrolysis gives amino acid. This is in terms of words, and this is by taking an example and we are explaining. And by this method, it is possible to synthesize uh, glycine, alanine, valine, leucine, aspartic acid. Okay. Uh, this is about the one method of synthesis of amino acids. So I will go to the next method. So next method is from the malonic acid. So what is malonic acid? CH2COOH. COOH is called malonic acid. Malonic acid is esterified. Then we can say that ethyl malonate. So here one can frame a question. Describe the synthesis of phenylalanine. Describe the synthesis of phenylalanine from malonic ester. So this is the malonic ester. So this malonic ester in presence of base. In presence of base means this base can obstruct this proton. Why this base obstruct this proton? Because this group is called methylene group, and this carbon is called methylene carbon, and this proton is called methylene proton. So usually this methylene proton is acidic in nature because this bond pair is involved in a keto-enol tautomerism. So because of that, mild base can obstruct this proton. Once the base obstruct this proton, these bond pairs are free. This bond pair can form a bond with this carbon and Cl will go out. So that is how an intermediate is obtained like this. So after obtaining this intermediate, what will happen? So when it is subjected to hydrolysis, hydrolysis means ester hydrolysis. You know that this is an ester. So there will be a cleavage here. So ester is converted into carboxylic acid when it is treated with KOH. KOH is a base hydrolysis. You can also take with the sodium hydroxide, barium hydroxide. So the product is hydrolysis. So the here the living group ETO and 1H. So ethanol will go out as a byproduct. Here also ethanol will go out as a byproduct. Formed one is called a acid. So more particularly diacid means dicarboxylic acid. So this one subjected to bromination. Where it will brominate? Again, I am telling you, this carbon is acidic in nature because this carbon is attached to two carboxylic acid. Two carboxylic acids are the electron withdrawing group. So because of that, this proton is acidic. That proton means it will form a new bond with the Br. So that is how halogenated to this carbon. The obtained compound is this. This obtained compound on heating, you know that any compound which contain two carboxylic acid to the same carbon atom, so that compound on heating undergoes decarboxylation. Okay, decarboxylation means carbon dioxide is going, this bond pair picks up a proton. So that is how a compound is obtained. So this compound on treating with ammonia. So this ammonia replaces Br. Okay, Br will go out, ammonia, it will form a new bond with this carbon. So the obtained compound is phenylalanine. Suppose in this method, if you want to prepare alanine, then what you need to take? Here you have to take methyl iodide. Then this bond pair attacks the methyl group, iodide will go out. Then in the alanine as the product. Suppose if you want to take isopropyl chloride, then you will end up with valine. Isobutyl chloride, then you will end up with isovaline. So this is how you can, just by changing the electrophile, you will get a desired amino acids. So now I discussed the two methods of synthesis of amino acids, and also I have discussed about 20 different amino acids, their structure and their symbol, and also I have given introduction about proteins and amino acids. So is there any doubt up to this?
No, sir.
Okay. So what I
Sir.
will do, yes,
No, sir.
what I will do at this stage, I will stop. Maybe I will continue in my next class. If you have any doubt, you can ask. Otherwise, I will stop presenting, and maybe you can log out.
Thank you, sir.
It's all right.